Thanks, Maya. And also thanks to everyone for sticking around. I'm going to try to keep it brief. So today I'm going to talk about heat tolerance and recovery capacity of corals from the naturally extreme Kimberley region. And in the beginning, I would just like to briefly acknowledge the many people and funding sources that have supported this research, but particularly also the Bardi-Javi indigenous ranges and traditional owners. We've heard it often enough today and yesterday. Coral reefs are struggling with climate change, and there is an urgent need to understand the mechanisms that may enable them to persist into the future and to acquire resistance to climate change and recurrent mass bleachings. And naturally extreme reef location can provide important insights into the biological mechanisms that facilitate survival and success under these suboptimal physical-chemical conditions. In particularly, the thermally extreme reef environments can play a key role here because they often harbor naturally heat-resistant coral populations, and they might play a key role in providing genetic rescue to populations maladapted to rising ocean temperatures. Now, the extreme reef environment that I have been interested in and studied quite extensively over the last few years is the Kimberley region in northwestern Australia. And what makes this region so extreme is that it has the world's largest tropical tides, with a tidal range of up to 10 or even 12 meters in some locations. And despite that, we find abundant and diverse coral reefs in the Kimberley. And so, because of these extreme tides, quite extreme strong environmental gradients occur over very small spatial scales. And I will show you in my talk today that this can have dramatic implications for the resilience of these corals during a natural bleaching event. So this is my study site, Shell Island in Signet Bay. And when you go there at high tide, there is nothing really that special about this place. But when the tide goes out and the water level is suddenly 8 meters lower, it's a very different place, and you get this mosaic of different habitats depending on tidal exposure. There is a subtidal environment that doesn't really get cut off from the main ocean, and because of that, the environmental conditions there are relatively moderate, with only up to a maximum of 3-degree daily temperature variation. The corals in the subtidal areas only very rarely get exposed to air, only during the most extreme spring low tides. And the maximum summer temperatures at this site are approximately 31 degrees Celsius. Now, the intertidal is a very different place. So these tide pools form at low tide, and they often get completely cut off from the main ocean, which means that you get this shallow body where water flow is quite stagnant. And that means that you can get very extreme temperature fluctuations, up to 7 degrees daily, and short-term maximum temperatures that are close to 40 degrees Celsius. And then on top of that, corals in these tide pools often get exposed to air for relatively long periods of times. And surprisingly, they don't show any visible signs of stress during these periods, usually. Now, what makes this such an interesting natural laboratory to study the mechanisms underlying coral heat tolerance is that these tide pools and the subtidal environments have the same mean temperatures, but obviously the variability of temperature and also other factors differs quite dramatically. And I'm going to talk to you in this talk. I'm going to show you how these differences in environmental conditions really impact heat tolerance and recovery capacity. So what we also know from experimental work that I did in the Kimberley about these different habitats is that the subtidal corals are more susceptible to heat stress than the intertidal corals. And this is despite the fact that they harbor the same symbiodinium types. So this highlights the strong role of these strong environmental gradients that really influence heat tolerance and, as you will see, recovery capacity of these Kimberley corals. And I'm going to talk about some of the physiological and genetic mechanisms today that contribute to these observed strong differences. Now, as you all know, the 2016 bleaching event in Australia did not just impact the Great Barrier Reef, but also coral reefs in other parts of Australia, especially in northwestern Australia, where temperatures were much warmer than usual due to the El Niño and marine heat wave. And at Shell Island, my study site, was obviously no exception, and so coral started to really experience heat stress already in the beginning of December 2015, and then maximum temperatures were recorded in March, April 2016, and they reached a maximum of about 6-degree heating wakes in both intertidal and subtidal environments during May. So we conducted coral health surveys in January, so 3 months before peak bleaching, during peak bleaching, then after 6 months of recovery. And here I'm going to show you the results from these in situ health surveys for both the intertidal and subtidal environments. And as you can see here on this PCA plot, for the subtidal is that the 3 different time points had very strong differences in the health in the community, in the health of the coral communities at Shell Island. So before the bleaching, the corals were dominated by unbleached healthy Acropora. That's the dominant coral genus in both the subtidal and intertidal. During the bleaching, it was dominated by severely bleached Acropora. And then unfortunately, 6 months later, the majority of these bleached corals had died. However, in the intertidal, it was quite a different story, although it started out quite similarly. So again, in the beginning, the health of the community was dominated by healthy and some Acropora and some healthy Porites as well. But there was also a really strong impact of bleaching on the communities in the intertidal, and the health of the community was dominated by mildly and severely bleached Acropora. However, remarkably, 6 months later, the majority of the bleached corals had fully recovered, and the difference between the pre- and post-bleaching health of the community was no longer statistically significant. So this was a dramatic difference in recovery capacity between intertidal and subtidal corals that really confirmed these known differences in heat tolerance. Now, of course, I was also interested as a physiologist in understanding how the physiology of intertidal and subtidal corals differed and enabled these strong differences in the recovery capacity of these corals. So, I wanted to highlight these results from the surveys just a little bit more on this slide. So this is the same data, but it shows them from the perspective of the different bleaching categories that we had, either healthy, mildly bleached, severely bleached, or dead. And as you can see, there was a pretty high percentage of severely bleached corals in both intertidal and subtidal habitats, even though there were more severely bleached corals in the subtidal. But because most of these severely bleached corals died, up to 70%, there was this huge drop in the cover of healthy corals in the subtidal, whereas in contrast, the amount of healthy corals in the intertidal essentially stayed the same. So now to understand the physiological mechanisms that were potentially contributing and underlying these strong differences in recovery capacity, we also tagged 10 healthy and 10 visibly bleached Acropora aspera colonies in both the intertidal and subtidal during peak bleaching, and then reassessed them after 7 months of recovery. And at both of the time points, samples were collected for physiological and genomic analysis. And I should point out here that the physiology work was primarily done by one of my students, Maria Young. So we measured a whole range of health variables, but I'm only going to show you 2 key ones today, one related to simian health and one related to host health. So on these graphs, you see the intertidal corals on the left within each panel and the subtidal corals on the right. And the black bars represent the corals that were visibly healthy during peak bleaching, whereas the gray bars represent the corals that were visibly bleached during April. And so looking at the simian density, you can immediately see oops, yeah, there we go. So the bleached subtidal corals had much greater losses in algal symbionts than the intertidal coral communities. So essentially, this confirmed the results from our surveys and also some of the other experimental work demonstrating that the subtidal corals have a lower heat resistance. The recovery, the corals were able to fully recover in terms of symbiont densities within 7 months, at least for the intertidal corals, but it was difficult to say for the subtidal corals because there was actually only one colony of the bleached subtidal corals that survived. So there is only an N of 1 here. Now, the second other key proxy that I want to show you here is the total energy reserves. And this is a very important health proxy because we know that high levels of coral energy reserves, which is the sum of lipid, protein, and carbohydrate in the coral tissue, high levels of these energy reserves promote resistance to and recovery from bleaching. So we kind of expected that intertidal and subtidal corals might have different levels of these energy reserves, or they might use them differently. And this is indeed what we found, but not exactly in the way that we expected it. So essentially, it was the intertidal corals that catabolized energy reserves during bleaching, and there was also no difference in the overall level of energy reserves between healthy intertidal and subtidal corals at this time point. And surprisingly, the subtidal corals were able to maintain their energy reserves during bleaching, so quite unexpected. Now, I think what happened here is that the ability of intertidal corals to catabolize their energy reserves and use them as alternative carbon sources during bleaching actually enabled them to survive and recover from this bleaching event, whereas the subtidal corals, for some reason, were unable to do that. In addition, I also think that the drop in energy reserves level in these bleached intertidal corals potentially reflects the cost of investing more in photoprotective mechanisms. So ultimately, it was this ability to catabolize energy reserves that enabled them to recover more quickly. Now, the physiology data again confirmed these strong differences in physiology between intertidal and subtidal corals. But I was also interested in finding out whether there is potentially a genetic difference between the intertidal and subtidal coral populations. And so this is something that I'm currently collaborating on with Dr. Luke Thomas from UWI and Ames in Perth. And the first question that we wanted to address was whether any cryptic genetic diversity in Acropora aspera might be confounding these strong differences between environments that we were seeing in the ecological and physiological data, because Acropora aspera is known to be a cryptic species complex. So Luke conducted low-coverage whole genome sequences of the same coral samples that we analyzed for the physiology. And we then found, indeed, that at our study site there is at least 3 different cryptic genetic lineages, 1 large one and 2 smaller ones. Interestingly, however, oh God, okay, no, the graphs are not there. Okay, so you will have to take my word on this. That's disappointing, but yeah, interestingly, the 3 genetic lineages all co-occurred in both the intertidal and subtidal environment in relatively similar proportions. And in addition to that, all 3 lineages had both visibly healthy and both visibly bleached corals during the bleaching event. So that already suggested that these different genetic lineages do not have a substantially different heat tolerance. And that was ultimately also confirmed by a permutational multivariate analysis of variance that I conducted across 9 coral health proxies. And that also showed that the physiology of these corals differs significantly between intertidal and subtidal environments, but not among the three cryptic genetic lineages. And the interaction between lineage and environment was not statistically significant. So this again highlighted that it is really the environmental gradients that drive differences between in heat tolerance and recovery capacity rather than cryptic genetic diversity itself. And the next step will now be to look at the potential genomic differences between intertidal and subtidal coral populations. So in summary, the 2016 bleaching event confirmed that strong environmental gradients can dramatically impact coral heat tolerance and recovery capacity over small spatial scales. The intertidal and subtidal habitats are really only separated by 100 or 200 meters. And then you also have to keep in mind that these strong environmental differences between the two environments only exist at low tide, but not at high tide. Surprisingly, the ability to maintain energy reserves during bleaching is not necessarily associated with high heat tolerance, and it does not guarantee survival. So this changes a little bit how we think about energy reserves and heat tolerance. And the co-occurrence of the three cryptic genetic lineages of Acropora asperi across strong environmental gradients highlights that acclimatization and their adaptation to these environmental gradients, rather than cryptic genetic diversity itself, drives the differential heat tolerance and recovery capacity of Kimberley corals. Thank you.